Hello, and welcome to Spotlight the Science, where we introduce and talk to some of our most interesting and amazing researchers that we fund at the American Diabetes Association. So, my name is Marlon Pragnell, and I'm the Vice President of Research and Science at the ADA. And it's really my great pleasure to have Dr. Shiubing Chen with us today. Dr. Chen is Associate Professor of Chemical Biology in Surgery at the Weill Cornell Medical College, and her major research interest is the manipulation of stem cell fate using chemical and biological approaches to generate functional tissues and organs that can be used for translational research. And her long term goal is to apply patient specific stem cell research derived tissues or organs for replacement therapy. And the goal is to build up a disease in a dish platforms for drug discovery. But first, what I would like to do is kind of talk to her more about the more recent research which she's been doing as well, and that we've been funding around COVID. Welcome, Dr. Chen.
Marlon, thank you. Thank you for giving me the opportunity to introduce our research in the spotlight of American Diabetes Association. Yeah, I'll be very lucky. Actually, I was a ADA Junior Faculty Awardee, and I also get the Innovative Award. And recently, we also get the support from American Diabetes Association on the COVID-related research. And it's kind of start from unexpected funding. As you mentioned, we are interested in set up this organoid platform. So we want to understand which type of virus, which type of cells can be infected by virus and how they respond to viral infection. And we started back to Zika, and then at the 2020, at the beginning of 2020, when New York was the pandemic, unfortunately was the pandemic center of the world, and we kind of shift our research focus to COVID. And at that time, we kind of originally we want to study lung and all small, large intestine because which believed to be the major target for SARS-CoV-2, the virus cause the COVID. And when we screen different type of cells, the organoid, very surprisingly, we find that pancreas beta cells, which is a cell that make insulin, and this is a hormone that maintains the blood glucose level, that can also be infected by SARS-CoV-2. I still remember that was the last experiment we did before the city was shut down, and I was completely surprised. And then we continue working on the project. At that time, we have a lot of patients in the ICU of Presbyterian Hospital. And we also have a lot of colleagues who serve in the ICU. And when sometimes we saw them in the hallway, we were just chatting, and they say, oh, you know what? We find it's very difficult to maintain their blood glucose level, some patients. And of course, the patient in the ICU, they are overnutrition. I mean, they have under a lot of control. But for the ICU doctor, they are very good to control their blood glucose level. So that is first time we thought there may be some clinical symptom which is related what we find in the experiment, in the lab, in the petri dish. So we continue working on that, and we collaborate with the lab in Japan as well as Dr. Francis Collins from NH. So we basically want to understand what happened to human islets once they got infected. And indeed, we find that after infection, we run an experiment called single cell RNA-seq. So we can monitor every single cells at the RNA level, how they change. And then we find that they lose the insulin expression. So they lose their beta, which is a major function of pancreatic beta cells. And very surprisingly, they also gain the expression of exocrine cell markers, like enzyme, like trypsin, which is actually the exocrine cells that secrete to digest the food. So we call this transdifferentiation process. So we think that upon SARS-CoV-2 infection, the beta cell, they might lose their identity and they change into other type of cells. So in that scenario, they cannot make enough insulin that may at least partially contribute to the difficulty to maintain blood glucose level in COVID patients.
Wow, that's so interesting. So you found that then infection of beta cells in vitro could actually drive transdifferentiation, make them function differently. So this then could be a potential reason why we're seeing an increased level of diabetes actually as a result of the pandemic, right?
Yeah, so I think that's we won't say it's the only reason, but I think it's definitely contribute to the increase the curve. And now we start to see as the 2020, 2021, we see some case reports of the new onset diabetes of the patient, COVID patient. And recently we start to see the population study. CDC just published a study say in the young adults and their increased rate of new onset diabetes.
So with these are actually people that are in terms of infectious. I know you looked at patient samples, people who have passed on as a result of COVID, and I gather these are people that didn't have diabetes, right? So these were people that just had COVID infection, and somehow you see these and actually the beta cells that you actually they try and differentiate it.
Yeah, so we think the interaction between COVID and diabetes is like we call it two-direction interaction. So if the patient they have like diabetes, sometimes they don't respond. If they got COVID infection, SARS-CoV-2 infection, they don't respond very well. And they have a higher risk to be hospitalized and to be sent to ICU. And in another way, and if for the patient we study, they are healthy. They don't really have they don't have obesity, they don't have diabetes before COVID infection. But after an infection, actually, when we take the pancreas autopsy, basically pancreatic tissues from the patient who passed away because of the COVID, and for some patients, we did directly detect the SARS-CoV-2 virus in the beta cells.
Wow! And if you look back at those patients that you did detect the virus and you saw the transdifferentiation, did you see those patients have clinical features of diabetes or impaired glucose control or so on, versus the ones that if you controls that didn't have infection?
So that's when we analyze the autopsy samples, the patient samples, we actually focus on the patients who have diabetes symptoms, so that there are pretty high correlation between the detection of SARS-CoV-2 virus as the patient who have hard response to like a glucose stimulation, who difficulty to maintain blood glucose levels.
Wow. And I understand you're an expert in how beta cells are created and from progenitor cells and so on. And you did some very nice work actually in the same paper that came out where you actually looked at actually the way that, or the mechanism that was causing this, and actually were able to kind of prevent it by some intervention there. I mean, obviously in terms of research and so on, but that was very interesting as well. Can you talk a bit about that?
Yeah. So I work on human stem cell model, and then to make pancreatic beta cells that can be used for replacement therapy and like disease modeling in the field. And then we actually when we make the pancreatic beta cells, it's a long work. I started working on that when I was a postdoc in Doc Melton's lab. So that's I've been working in the field for almost like 15 years. So, and it's good. You actually see it's a stepwise procedure. So you start from human pluripotent stem cells, and you go to affinity endoderm, pancreatic progenitor, and then to beta cells. And every step, we need to give them the right signal at the right timing. So then they will convert it. And the field really moved forward a lot. I still remember the first time when I decided to join the field when I was a graduate student in 2004, and I heard a talk from Dr. Melton, and he basically at that time it's still a dream or a really ambitious goal to make beta cells for replacement therapy for type 2 diabetes patients. I said, oh, that is something I want to do. And now if you think back, like almost 20 years later, we start to put these beta cells into patients, and then to see very nice response, and which is really exciting.
It's amazing. I've been following it. It's absolutely incredibly exciting because the challenge with replacing beta cells, they were able to do beta cell transplants from patients who who had passed on, and there's evidence, yeah, that you can actually replace beta cells. And there's even, I think, it's even one person that's kind of, it worked really well, where he had a transplant and was no insulin dependence for many, many years. And so now this actually is almost like we're on the threshold of some really interesting developments here, where we're seeing the potential for actually creating beta cells in culture by using some of the techniques that you're using, for example. And I know from Doug Melton's lab, where you've take cells and actually coax them, give them little signals at different stages to actually take them into the stage where they're a beta cell that you could potentially transfer into a person. So potentially this is like unlimited amounts of beta cells that you could create, right?
That is a goal. That is a goal, and I think that's I still remember when we're describing, we can have like a cell that can fill the room that can use in the patient. But I think that's a major advantage for stem cell, because stem cell in theory they have unlimited proliferation ability. So we have a limit starting material to make the type of cells we need. And so another advantage for stem cell is that, well, immuno attack is a major concern for type 1 diabetes patients. And in stem cells right now, including my lab and some other labs, they are kind of doing some manipulation on the stem cells so that to make the stem cell can be invisible, so that they won't be attacked by the immune system. So that can be like a next generation cells that can be used for transplantation therapy. Yeah.
Wow. Because that's the main issue, right? With type 1 diabetes, for example, where you have transplanted cells, but then individuals have to kind of undergo kind of immune suppression therapy, which is kind of challenging. So if you were able to find a way that you could actually generate large amounts of beta cells in culture, and then also to find a way that they are not perceived as foreign by the body, so they're accepted and then grafted and function, that would be amazing to accomplish that.
Exactly. That's actually one major direction we are trying to, I mean, many labs in the field are trying to pursue to use for cell replacement therapy. And in the meanwhile, we can also use the cells we derive from the petri dish for other purpose. For example, we run a lot of chemical screening, and in many type 1 diabetes patients, even very late stage type 2 diabetes patients, they still have some residue beta cells. So they still have beta cell left. And if we can just simplify, double the number of these where their beta cell, triple the number of their residue beta cells, they might have enough insulin to maintain the blood glucose level. So beta cell proliferation is also another field that's very active. And then we are running a lot of drug screening to find this. So using the stem cell derived cells, which we have very robust supply to run the screening to find the small molecule that can promote beta cell proliferation. And another chemical screening we are running right now is in type 2 diabetes, the beta cell goal transdifferentiation, just as similar case as COVID patients. So they basically lose insulin, they basically gain the other cell identity, and we start to run the screening. We find the small molecule. If we block the EF pathway, there are some candidate small molecule, they can block the transdifferentiation process. So even though we are really under stress, but they still can maintain their identity. They are still beta cells, and then they can still maintain their identity and then still be functional.
Wow. Yeah, because people with type 1 diabetes, for example, they do have residual beta cells, right? And so if you can find a way, a strategy, an approach to kind of coax those to replicate, to create more of themselves, you could then bring back the population of beta cells that would be potentially enough to provide the insulin that you need to be insulin independent, right?
Yeah, so actually there are already clinical trials. They basically try to use a small molecule to promote beta cell proliferation, and we are interested to see whether we can find additional drug candidates.
So you're looking screening libraries. So you look at this in the culture in the laboratory in the petri dish. These large screens of like libraries of different compounds, and then you see then can these the ones that jump out at you think of those maybe in a clinical trial where maybe people take a drug, for example, or that might actually specifically target their beta cells and kind of cause them to kind of replicate. Is that right?
Yeah, exactly. So the drug we are screening is it's a library that's include the drugs either already approved either by FDA or Europe country, or it's drugs in phase 1 to phase 3 clinical.
Wow.
So this small molecule, there's really drug. They have already been validated in the animal. Their PK/PD is good, so we can directly repurpose this drug
Wow.
for
Wow.
the diabetes patient. That's the goal.
Wow. This is incredibly fascinating, and one of the area that I think you're also working on is incredibly fascinating. This idea of occurring a disease in a dish, you know, with an organoid from potentially a individual that has a certain disease or some issue and so on. You can actually create an organoid from their tissue, right? And actually look and see screen drugs and see how you could potentially treat that or identify drugs that might be of benefit there. Can you talk a bit more about that? I find that incredibly fascinating.
Thank you, Maureen. Thank you for mentioning that. We actually, for now, 2019, we set up a diabetes program. So we currently have a large IRB protocol. Basically, it's a collaboration with Illumina. So we will collect samples from 500 diabetes patients, and we will perform genomic sequencing, whole genome sequencing for all these patients. In the meanwhile, we will make stem cells from these patients, and then we can make beta cells from these patients. And so it's basically we start from the whole genome sequence. We will and then we study their beta cells. So we'll do this kind of phenotype to genotype correlation study, and the goal is we want to find what's the key genetic factor that causes beta cell failure, like beta cell dysfunction or like beta cell death, and then we can do this target therapy. For
Yeah.
example, we find when we work on a gene called GLS3, which is GLIA3, is involved in both type 1 and type 2 diabetes. And when we run the drug screening, we find the TGL beta inhibitor, which can basically protect this particular type of beta cells.
Wow.
So in that scenario, that's more like for the personalized medicine or precision medicine ago. So people may get diabetes because of different cause. And right now, many patients that were treated using the same approach. So based on their genetic background, and we might be able to basically separate patients into different groups. And the goal is we want to develop like target therapy for each group.
Wow. So you're saying then, in a sense, that people develop diabetes, whether type 1 or type 2, potentially because of a predisposition as a result of their genetics and so on. And so when you do this work, you are actually recruiting cells from them, collecting cells from them, but also you're doing the genetic analysis. And so then what you're putting two and two together, and then potentially seeing those individuals that develop diabetes in a certain way, then you can actually identify what's the approach, what therapy, drug-based approach you can use to kind of stop that or reverse that. Is that right?
Exactly. So I can give you an example. Recently, we make stem cells from around 100 patients, and then we make beta cell from these patients. And very surprisingly, or maybe not surprising, so when we make the beta cell, we expose them to like we give them high glucose and high fatty acid, basically to mimic high glucose, high blood sugars, or like a high fat obesity condition. And indeed, we find some patient beta cell, they are very strong. So you expose them, they survive. Okay. Some patient beta cell, they are very fragile. So even though you give them a little bit challenge, the cell die. And now then we go back to check their genetic data, and then we find some genes that actually play a very important role for the beta cell death. So then we basically for this type of patient in the future, and if the patient carry this genetic defect, it doesn't mean the subject will directly immediately get diabetes. At least I think this patient will be on high risk for diabetes. For example, if they got obese, most likely their beta cell won't be able to handle the stress very well. So for this type of patient, our goal is once we find the genetic factor, we want to find the drug, or at least drug candidates that can protect the beta cell.
Fascinating. So, because we do know that diabetes is quite heterogeneous and people have different levels of susceptibility and so on, and there may be different genes that might kind of predispose people. And so, I mean, your approach there, in a sense, to create more of a personalized strategy where you can identify and potentially kind of identify subsets of individuals where you might have a more effective approach to prevent or treat their diabetes, right?
So that's specific goal because we all know that a different lot of genetic factor contribute to type 2 diabetes. And so and I think if one solution, even though we have different drugs, but many of the drugs is still trying to solve the problem, either directly decrease the blood glucose level. So and as we discussed before that many of them they still have beta cells. And if we can promote their beta cell function, if we can promote their beta cell proliferation, if we can protect their beta cells, and maybe we can solve the problem from the model.
Yeah, I think that's why research is so exciting now. I mean, there's so much progress and so much momentum towards, uh, you know, identifying specific patient types, also to strategies to restore beta cells, whether people's beta cells kind of expanding them, the ones they have there to kind of make more of them, or potentially, for example, as you mentioned as well, like developing them and creating huge quantities in the laboratory, so then you could actually provide individuals with beta cells and insulin dependence. Absolutely fascinating. And then also too, I mean, going back to what we discussed earlier, I mean, when you discuss also about the infection of beta cells with COVID and how that we see the transdifferentiation. That's interesting as well because that also helps you understand what all the different pathways that occur, all the activity inside the beta cell. That adds to your knowledge and understanding of how the beta cells work. And who knows, maybe from there we could actually add to the knowledge of beta cell function and differentiation and identify drugs and so on that could potentially work in different ways to protect people or treat people who have diabetes.
Exactly. I think that's the value of the beautiful part of the basic science. I think we really need to understand what happened and then we can cure the disease.
Very exciting. I think this is one of the most, I feel this is probably one of the most exciting and interesting areas of research right now in the world in terms of diabetes. Understanding the beta cell, being able to produce large quantities of beta cells for transplantation, strategies to reproduce or to expand the existing very small beta cell population that people may have, residual levels, and so absolutely fascinating. It's a pleasure and a privilege for ADA to be able to support really brilliant researchers like you in the field. And really thank you for all the work you're doing towards this effort that we have to go to cure, manage, and prevent diabetes. This is such an important area of research that you're pursuing. Really appreciate your efforts here.
I really want to thank ADA for the support of my career and our research. And it's really helped us from the beginning when I set up the lab to basically build up the stem cell model and like to build up the disease model and then go to different like genetic factors and now go to COVID. So we really, with the support from ADA, we actually set up multiple models we can use to systematically study how different genetic factor, different environmental factors like virus, how they contribute to diabetes progression. And with these all new models, we adapted to drug screening platform and then look for new drugs.
Definitely. This is exactly what ADA is interested in doing, obviously, and we want to support the best research to help people with diabetes to find therapies to better manage the disease, to better prevent the disease, cure the disease. And I think this is one of the most interesting areas. Thank you so much. I know we're almost at time now, but really appreciate your time today, and we look forward to some really nice, interesting work from you in the future. Thanks so much, Dr. Shiubing Chen.
Thank you. Thank you. It's really nice to talk with you. And thank you for the opportunity to introduce our research.
Thank you very much.